This video is going to be all about the nephron. The nephron is basically the functional unit of a kidney. So when we talk about all of the wonderful things that the kidney does, we're basically talking about what's happening at the nephron. This is a diagram of what a single nephron looks like. Nephrons are located in the cortex of the kidney, so in there. And there are a ton of them in a single kidney, about 1 million. The nephron starts here at the glomerular capsule, extends through the proximal convoluted tubule, through the descending limb of the loop of Henle, and the ascending limb, then through the distal convoluted tubule, and the nephron empties into a collecting duct. And each collecting duct takes urine from multiple nephrons. So that's what each of these little extensions are places where urine from a different nephron enters into the collecting duct. The collecting ducts aggregate into papillary ducts, which deliver urine down to the minor calyx. From the minor calyx, that urine then passes to the major calyx, the renal pelvis, down into the ureter and the bladder. A few of these structures of the nephron have different names that you should also be familiar with. The glomerular capsule is sometimes referred to as the Bowman's capsule, and the loop of Henle is sometimes referred to as the nephron loop. Lastly, the collecting duct. Some texts will call it the collecting system. You should be familiar with both terms for each structure. The glomerular capsule is part of a structure referred to as the renal corpuscle, and that just includes this little knot of capillaries called the glomerulus. The glomerular capsule wraps around the glomerulus like a close-fitting cup. These arrows in the nephron indicate the direction that urine flows as it passes from the glomerular capsule, where the process of urine formation begins, through the PCT, through the nephron loop, the DCT, and into the collecting duct. There are 3 processes that occur along the nephron in order to make urine. The first is glomerular filtration. As you might guess from the name, glomerular filtration happens at the renal corpuscle. Glomerular filtration is basically the process of the blood pressure in this knot of capillaries, the glomerulus. It's a bit of an unusual capillary bed because it actually has a fairly high blood pressure. So the pressure of the blood in these capillary beds forces plasma out of the capillaries into the space of the glomerular capsule. This is the first step in making urine. So urine really starts with plasma. The second step in making urine is tubular reabsorption. In tubular reabsorption, the kidneys reclaim almost everything from the plasma. Basically, tubular reabsorption allows your body to hold on to things that it doesn't want to get rid of. Things like nutrients, vitamins, glucose, amino acids, certain ions, water, and the rest, the things that aren't reabsorbed, continue out of your body in the urine. The proximal convoluted tubule is the main location of tubular reabsorption. That's where most tubular reabsorption occurs, but there is reabsorption in the nephron loop, distal convoluted tubule, and the collecting duct as well. The third process in urine formation is tubular secretion. Tubular secretion is where your kidneys take items that either weren't in the filtrate or items that had been reabsorbed and secretes them back into the urine. So it's moving substances out of the interstitial fluid back into the urine. And tubular secretion is used to sort of fine-tune the composition of the urine. It's also involved in regulating the acid-base composition of the blood, tubular secretion is a large part of how the kidneys help out with that. And tubular secretion happens all along the nephron at the proximal convoluted tubule, distal convoluted tubule, and the collecting duct. So now I want to go into a little bit more detail about the different regions of the nephron. The first region, as I mentioned, is the renal corpuscle, and this is where you get glomerular filtration happening. Blood plasma filtered out of the glomerular capillaries into the lumen of the renal tubules. And actually, it goes directly into this inner space of the glomerular capsule. So the glomerulus itself is the capillary bed. It's high pressure. It's fenestrated, which means that it has a number of openings in it, making it a very permeable capillary bed as well. The glomerular capsule is a two-layered cup, so there is a visceral layer that wraps tightly around the capillaries of the glomerulus, and a parietal layer of simple squamous epithelium. There's also a little bit of space in between the two, and this space is the capsular space. This is the space that urine enters into, and this is the space that's continuous with the proximal convoluted tubule. Here's another look at the renal corpuscle under the microscope with the different structures highlighted. So you have this is actually the parietal layer of the glomerular capsule highlighted, the capsular space, and then the glomerulus itself. You can actually see little red blood cells in those capillaries of the glomerulus. Now, the combination of the endothelium of the capillaries and the visceral layer of the glomerular capsule, that's what determines what enters into filtrate. So that's what determines what passes into the nephron in that first step of urine formation, is the structure of this membrane. And it's referred to as the filtration membrane. If you could zoom in super close on a single capillary of the glomerulus, this is what you'd see. You can see the capillary itself under here, and you can see the fenestrations, the pores or openings in the endothelium of the capillary. These cells, the podocytes, are the cells that make up the visceral layer of the glomerular capsule. These podocytes are highly specialized epithelial cells with numerous cytoplasmic extensions, and these cytoplasmic extensions form openings called filtration slits. And urine, or not urine yet, but plasma, as it's becoming filtrate, passes through these filtration slits. So the filtration membrane is basically the barrier across which plasma is turned into filtrates. That filtrate that will become urine as it passes through the nephron. And there are three components to the filtration membrane. You have the capillary endothelium, so that's this stuff. And remember, it's got the fenestrations, it's got the pores, so it's very permeable. You also have the basement membrane, which is that little bit of connective tissue between the endothelium and the podocytes. And the third component is the foot processes of the podocytes. These foot processes are these little sort of extensions of the cytoplasmic extensions. Some textbooks refer to this basement membrane as the dense layer. So as you have blood passing through the glomerular capillary under high pressure, that pressure forces blood out of the fenestrations in the capillary endothelium, through the basement membrane, through the filtration slits, into the capsular space, the space between the podocytes and the parietal layer of the glomerular capsule. But not everything goes through. This is a filtration process, right? So some things stay in the capillary, and only some things make it across this barrier, across the filtration membrane, into the capsular space. So although the endothelium and the podocytes are very porous, things that are protein-sized or bigger don't cross, right? So typically, there's some variation in this, but typically protein and cells in the blood aren't found in the filtrate. They're excluded by glomerular filtration. This is another view at that filtration membrane. This is an actual scanning electron micrograph of the glomerular capillaries. The capillaries are underneath here, and you can see the podocytes sitting wrapped around the capillaries. The podocyte cell body with its cytoplasmic extensions, and the foot processes coming off of those cytoplasmic extensions, and then the filtration slits in between the foot processes. Foot processes are also sometimes called pedicels. Podocytes are weird looking. They're probably my second favorite cell in the human body, just for sheer bizarreness, after the osteoclasts, of course. We'll talk a lot more about the processes of glomerular filtration and tubular reabsorption and tubular secretion when we get into the physiology video. I do want to introduce some of the structure of the rest of the nephron here, including thinking about the histology of the nephron. In the proximal convoluted tubule, you have cuboidal epithelium with large mitochondria and dense microvilli. Remember that I said the proximal convoluted tubule is where the majority of reabsorption happens? Well, just like in the small intestine, the microvilli of the proximal convoluted tubule increase the surface area so that water and electrolytes and nutrients can be reabsorbed into the blood. In the loop of Henle, you have both a thick segment and a thin segment. The thick segment has cuboidal epithelium and is less permeable to water. The thin segment has a simple squamous epithelium, so it's easier for substances to cross, and it is water permeable. When we start talking about urine concentration, this distinction between water permeable and water impermeable epithelium will become important. In the distal convoluted tubule, you're back to cuboidal epithelium, but this cuboidal epithelium, there's still some reabsorption going on, but not as much, so there's actually no microvilli in the distal convoluted tubule. These transmission electron micrographs show you the differences in the cells of the proximal convoluted tubule versus the distal convoluted tubule. The proximal convoluted tubule, here are your microvilli, and you can also see how dark staining the cells are with mitochondria. In the distal convoluted tubule, no microvilli. From the distal convoluted tubule, urine passes into the collecting ducts. There are two main types of cells in the collecting ducts. They're not that easy to distinguish histologically, and that's not really the most important point I want to make. I want to distinguish between them in terms of function. There are intercalated cells and principal cells. And the intercalated cells are primarily involved in maintaining acid-base balance of the blood. These are the cells that secrete hydrogen ions into the urine when your blood is too acidic. So they get rid of some of those excess hydrogen ions by sending them out in your urine. The principal cells are mostly important to maintaining the water-sodium balance. When we talk about the effects of aldosterone on urine formation, aldosterone places a sodium-potassium ATPase in these principal cells so that sodium gets reabsorbed and potassium gets excreted. When we talk about antidiuretic hormone triggering the increased reabsorption of water, out of the urine, it works by acting on the principal cells. Antidiuretic hormone places aquaporins, water channels, in the membrane of the principal cells so that water can be reabsorbed out of the urine back into the body. This slide and the next slide serve as a summary of the different cell types you find along the nephron. So we've got the simple squamous cells of the parietal layer of the glomerular capsule, and that's just purely structural. It's just the wall. It just holds the filtrate in the capsular space. You have the podocytes of the visceral layer of the glomerular capsule with their specialized foot processes that form filtration slits, and these guys are actually involved in the filtration process. The structure and integrity of these podocytes is important in controlling what goes into that filtrate. In the proximal convoluted tubule, you have cuboidal cells with lots of mitochondria to make ATP to drive active reabsorption, and dense microvilli to increase the surface area for reabsorption and secretion. In the distal convoluted tubule, you have cuboidal cells but no microvilli because there's a lot less reabsorption here. In the loop of Henle, you have thin segment cells that are simple squamous and water permeable, and the thick segment cells are cuboidal. Lastly, in the collecting duct, you have both the principal cells and the intercalated cells. Now, I've been talking about the structure of a nephron in general, but I do want to point out that there are actually two types of nephrons, cortical nephrons and juxtamedullary nephrons. Most of the nephrons in your kidney are cortical. About 85% of your nephrons are cortical nephrons. And these guys are almost entirely in the cortex of the kidney, except for just a very small part. The deepest part of the loop of Henle is located in the medulla. Juxtamedullary nephrons, as you might guess from the name, are located at the junction between the cortex and the medulla. So they're right at the border between those two regions of the kidney. And the loops of Henle are much longer and go much deeper into the medulla. There are some differences in the paratubular capillaries of these two as well. So cortical nephrons, they have paratubular capillaries wrapping around their proximal and distal convoluted tubules and around the loop of Henle. Juxtamedullary nephrons have paratubular capillaries around their proximal and distal convoluted tubules, but they have a capillary bed that wraps around the loop of Henle that is much more structured, parallel, and geometrical, and this is called the vasa recta, so the straight vasculature. The long loop of Henle and the straight parallel flowing vasa recta are important in the formation of concentrated urine. Having this very deep loop of Henle allows the juxtamedullary nephrons to set up a steep concentration gradient so that the solutes down here in the medulla are there's a very high solute concentration in the interstitial fluid down here around the juxtamedullary loops of Henle. The vasa recta, the arrangement of the vasa recta, allows for return of electrolytes and water to the blood without disrupting that steep concentration gradient. And as we'll talk a little bit more about in another video, that that high solute concentration down here means that when you have water, that means that you get more water being drawn out of the collecting duct, basically, through osmosis. That high solute concentration sucks water out of the collecting duct so that you don't get seriously dehydrated because you're losing too much water in your urine. So when we're talking about the vasculature around the nephron, we really have to consider the glomerulus and the peritubular capillaries and vasa recta separately. They are very different types of capillaries. The glomerulus is specialized for glomerular filtration. It's highly pressurized in order to push plasma out of the capillary into the nephron. And it's a bit unusual as well. If you think back to the cardiovascular system, capillary beds are usually fed by an arteriole and drained by a venule. But the glomerulus is actually fed and drained by arterioles. The afferent arteriole brings blood into the glomerulus, and the efferent or efferent arteriole takes blood out of the glomerulus. Blood flows from the efferent arteriole into the peritubular capillaries, and the peritubular capillaries are a more normal capillary bed, very low pressure, drained by venules. And these guys are closely wrapped around the renal tubules, and they're also highly porous, so that substances that have left the tubules, entered into the interstitial space, can be taken back up into the peritubular capillaries and returned to circulation. The vasa recta, similar to the peritubular capillary beds, low pressure, porous, but slightly more linear in structure and tightly associated with the loop of Henle. So we focused on structure in this video more than physiology, more than process, but you should still be able to talk a little bit about how the structures discussed in this video relate to the processes. You may even want to come back and revisit this video after familiarizing yourself better with the processes of urine formation. Regardless whether you do that or not, after studying this video, you should be able to talk about the anatomy of the nephron, the structure of the filtration membrane, at least introduce the processes that happen at the renal corpuscle and in the renal tubule. Talk a little bit about the difference in function between the intercalated and principal cells of the collecting ducts, and talk about what's going on at the capillary beds of the nephron, the glomerulus and the peritubular capillaries.